Hello, everyone, and welcome to our Friday seminars. It is my pleasure to introduce to you today our speaker, Dr. Michael Love from UNC Chapel Hill. Michael is an associate professor there, jointly with the Department of Biostatistics and Department of Genetics. He received his PhD in computational biology from Prague University in Berlin. Hope I got that right.
Perfect.
And he also then completed a postdoc at Dana-Farber Cancer Institute in Harvard before joining UNC. He has a, needless to say, he has a very extensive and well-established, research profile, and also a very well-functioning lab. Lots of members and doing lots of good quality work, high-impact work, appearing in top bioinformatics journals. His research interests generally are in developing statistical and computational methods for analyzing high throughput and high dimensional sequencing data with applications in understanding important challenges and important questions in modern biomedical research. And his research is also very well funded through multiple NIH grants, and his lab also develops multiple open source software for the bioconductor project, among other things. So today he's going to tell us about some of his recent interesting work on statistical challenges in the analysis of tumor subtypes for the Carolina breast cancer study. So please join me in welcoming Michael.
Hi, thanks everyone, and thanks again for inviting me. This has been a fantastic visit. Lots of shared research interests. So for today, I have a set of slides that go over mostly. I start with the data, like what kind of data this study has generated and what are the kind of statistical considerations. We have some, I think like pointers, like how will we figure out how to work with the data, but there's also a lot of remaining questions. So it's not a bunch of solutions, but maybe some open problems. And everything here is in collaboration with these two labs. So Melissa Troster in epidemiology and Katie Hoadley in computational medicine. And they're also members of the Lineberger Comprehensive Cancer Center. So kind of, these are my collaborators who introduced me to these data sets and to these problems I'm going to talk about today. And also we have a Center for Environmental Health and Susceptibility, which Melissa Troster is the director of. So I want to introduce the study first of all. So here I have the total number of participants in a 2x2 grid. So there's women with breast cancer enrolled in this study. There's multiple phases, and this focuses just on the most recent phase, which was 2008 to 2013. And as you can see, the sampling has designed on purpose to kind of equally represent Black women with breast cancer. In this, we have like non-Black is the other category here for self-reported race. It's predominantly women who self-identify as white. So that's the majority of the non-Black category. And then there's also divisions by age. So 20 years to 49 years or 50 plus. And just to note, so we have a 1-to-1 ratio of in both comparisons, and other public data sets of genomic data for breast cancer are much more skewed to women who self-report as white. So TCGA has a 5-to-1 ratio, and average age skews to the higher. So the study design purposefully upweights here women with breast cancer who are Black and younger as well. And the reason for that is these are the cases where we see more aggressive breast cancer. So this is recurrence in the third phase, and you can see the younger category as well as Black compared to non-Black women. You have higher rates of recurrence. These are more aggressive tumors, and we want to understand why. So the other thing that's kind of a confounder here is that the stage of the tumor at recruitment varies by age and race. So here we have the stage is this like red to blue coloring, and on the left hand side you can see we have the mode is stage 2 with a almost equal amount of stage 1 and stage 3 for young Black women. And if you move all the way to the right side, the older non-Black women, it's predominantly stage 1. So at recruitment, we're seeing higher stage, both for Black and young women in the study. So the study was initiated a while ago. So phase 1, the paper describing the study goes back to 1995. And this is like, I went back and read, when I started working on this study, I went back and read the paper that was describing the outset of the study. And there's some important things here I want to highlight. So from the very beginning, they were interested in breast cancer heterogeneity. So the idea that there's different types of, it's not a single monolithic disease, but there's subtypes. And they pointed out that the evidence of this is coming from multiple angles. So we see different risk factor profiles among women. We see racial disparities in incidence. And we also know that there's a small portion of breast cancer where there's, we can kind of explain it through family-based, there's some genetic predisposition to certain types of breast cancer. So these all contribute to describing there's some underlying but unknown etiologic heterogeneity. Etiologic meaning the causes of incidence of the tumor are heterogeneous. So it's not as if all women with breast cancer have the same set of causes, but it seems to cluster essentially different types of disease with different causes. And I just want to highlight, I think when we think about survival, in particular, racial disparities related to survival, we know now that, I mean, as we knew in this paper, but we emphasize now the differential access to treatment and, systematic racism that plays into the differences in treatment that will be offered to, women based on, so underlying racial disparities. Just a couple more, things I like to highlight from this original paper. So they already had identified potential mechanisms underlying the heterogeneity. So, genes for early onset, BRCA had just been identified, BRCA1 and 2, as well as seeing that overexpression of HER2, a gene, and overexpression of the gene P53 were explaining some variation in etiology and in outcomes. So these you could talk about as molecular subtypes. So when you examine the tumor, you examine the modification of the genome, you examine the expression of the genes, and you could start to subtype and try to connect differences in etiology to differences at the molecular level of the tumor. And those, since 1995, those are a routine part of treatment. So now we profile the tumor, and in many ways, the molecular subtype will dictate treatment, whether you deal with a hormone-positive tumor in one way, all the way to like immunotherapy. Like it will all be, informed by molecular subtyping. So this is my last. There's still more to gain from this this initial publication. So the goal of the study was to find distinct causal pathways, both at the individual and population level, and to try to identify molecular signatures that can help us understand the heterogeneity of the disease. So just to show, this is now jumping ahead 14 years, to show these are now, there's a researcher at UNC, Chuck Peru, who, oops, maybe I should make sure that's plugged in. A researcher at UNC, Chuck Peru, who did work in the early 2000s to identify gene expression-based subtypes of breast cancer. And this is to show, so these are Now these survival curves are, let me describe. On the left side is no adjuvant systemic therapy. So after primary treatment, not following up with other treatments to try to reduce the level of tumor cells down to zero, which is not common, but that's just to say that's trying to show you maybe aside from adjuvant treatment, what outcomes might look like. It's you can't run a study where you don't apply treatment, but this is no systemic therapy after the primary therapy, just to show you what survival curves look like. They obviously differ by these molecular subtypes. So I won't describe them in detail, but you can see luminal A has the best survival on the left-hand side, the blue curve, and then you have more aggressive subtypes, luminal B, HER2 enriched, and basal light. And then you can also see now this is in the next two plots are just stratifying patients by ER positive status. We know ER positive is kind of one of the most defining features of breast cancer, and you can see big differences between the way these curves follow for ER positive and ER negative tumors. So in particular, you can see luminal B goes from having almost some of the worst outcomes in ER positive to up towards luminal A in the hormone negative, ER negative case. So the point here I just want to show is there are big differences in outcomes based on this molecular subtyping, which you can do at the initial recruitment of a patient. You can oh, are we not on?
It stopped sharing the screen.
Oh, okay.
I'm so sorry.
That's fine. Yeah. I wonder why I did that. Anyway, okay. And then, okay, so I've showed so far you can subtype breast cancer tumors. That's relevant for survival outcomes. It kind of there's maybe additional subtyping, and then this is to show that incidence of these different types of tumor. So here I'm categorizing tumors by hormone receptor positive or negative and HER2 positive or negative. That forms 4 groups. And this is to show that this is a study of women from Nigeria compared to Black and white American women populations. And this is to show that there seems to be, at least at incidence, an ancestry component. So women in Nigeria compared to African American women compared to women self-reporting as white. There's seems to be subtyping has perhaps ancestry plays some role in the incidence of these different subtypes. And this is on the right-hand side now, a different subtyping. This is the one that you saw before, which was the basal type, HER2 type, and the 2 luminal types. And so again, there seems, for example, a basal, which some of these are triple negative breast cancer is very aggressive. They tend to you present more often in women with African ancestry. And at the bottom, there's an important caveat that the Nigerian women were much younger, had more advanced stage at diagnosis, which may reflect lack of screening and access to healthcare as well. So that was all just to kind of set up the background of breast cancer and heterogeneity of disease. And now I want to just list a bunch of statistical problems and considerations when we start working with this type of data. So, I mean, this goes back to, you mentioned, I trained at Dana-Farber with Rafa Irizarry. So this is one of his, most important topics. He's hammered into the community of statisticians who approach genomic data. There are these things called batch effects, which are basically technical distortions of the data, of the measurements that have nothing to do with the underlying biology, but have to do with the phase of the data, the time that the sample was processed. They're just correlations essentially in the data that are not due to biology, but just due to measurement. problems, so correlated measurement error. And those are going to vary by phase. And so we need to take that into consideration before we just approach the data and start using it naively. We often make bulk genomic measurements, which means take a tumor and measure the RNA in the whole tumor. We're moving now towards single cell and spatial measurements of tumors, but a key consideration whenever you have this kind of bulk data is that you're both seeing changes in expression in different types of cells, and that's confounded with different proportions of different types of cells. And so that's important because if you just, again, like naively compare two samples, you're saying, comparing like the measure, the number of RNA molecules, you're both mixing together the different types of cells, like epithelial cells and immune cells and tumor cells. You're mixing together their change in expression level within a cell type, as well as the different proportions of the cell type. When we look at risk for diseases based on genotype, so we can look at genome-wide association studies, we have to consider the fact that those heavily have been skewed towards European ancestry. There's an effort to correct this, but if you download a data set of genotype association with incidence of breast cancer, you have to take into account that that's going to be skewed towards women with European ancestry. It's very difficult to isolate the effects of societal injustice and access to healthcare. For example, the later stage and higher graded recruitment with effects from ancestry. So there's probably a combination of both, and it's hard to disentangle those two. This is a study. We've observed a number of variables. There can be confounding between variables that we want to control for with exposures. Say, if we want to look to see how different exposures affect someone's risk, we have to be concerned about confounding, and how we choose to build those, the directed acyclic graphs will matter. We can't just kind of naively loop through the variables and put one at the end and have that be the treatment variable or exposure variable. So questions like, how do we control for parity when contraceptive use is the risk factor of interest? Those both actually influence incidence of breast cancer. So how to do something like controlling for one when they might also be related to each other. And another thing, this is probably impossible because treatment is directed by a subtype. How do you possibly disentangle the effects of subtype on outcomes versus treatment? And I showed you this plot where no adjuvant systemic therapy. That's one way, but that's not very representative because that's not recommended treatment necessarily. So that may just not be possible given how much treatment is dictated by a subtype. So this is a summary slide of all the types of data we have in this study. We don't look at it all at one time. I'll show you some projects where we looked at pieces of all this data, but let me just walk through what we have. So we have risk factors and exposures for most women in the study. So oral contraceptive use, smoking, risk factors like this. We have information about treatment, and then we have outcomes or recurrence. We have, and so it's not a complete data matrix, we have some, missing data, but we have for many women, their germline DNA. So not the tumor DNA, but the, DNA from the blood. And so sometimes women have risk for breast cancer due to their, germline mutation, like BRCA1 mutant status, one copy of BRCA1 mutated. And that would be a family-based risk for breast cancer incidence. We also have the somatic DNA. So this is This is looking at the pattern of mutation that occurs in cells throughout the life. So I'm drawing these like green stars and blue stars. These are just mutations that occur in somatic cells over time. And those can tell you also about exposures that are not recorded. So for example, UV radiation or smoking induce certain patterns of mutations, and I'll show what that looks like in the second part. And then after, once at the incidence of cancer, additional mutations occur, and like I've drawn these orange stars. So these are like additional mutations that are kind of specific to a cell having gone down the path towards a tumor. So we can also look at, the mutated gene status in the tumor, which is different from the germline mutations. So an inherited mutation of an oncogene versus a acquired mutation during the oncogenesis. And we measure the RNA of the tumor, and we get this RNA-based subtype. Those are the ones I've been showing you, like basal, HER2-enriched luminal A, luminal B. So we measure that from the RNA in the tumor. I won't talk about it today, but in a subset, we also, if there is recurrence and if there are metastatic lesions, we can measure omics of the metastasis. External data sets include large-scale genome-wide association studies for incidence and survival, and measurements of expression in other tissues in other studies. So those are things we might use also. So the first thing I want to talk about is trying to associate germline DNA with breast cancer outcomes. The question here is, is there a heritable factor that explains why some women have recurrence and others do not? So we're going to focus on the germline DNA and outcomes, and we may also use the tumor subtype and the measurements of RNA in the tumor. So we're going to try to, the idea here basically is, instead of just doing an association for every position in the genome, we're going to use information about the tumor RNA to try to collapse the information from the genotype to units, which are based on genes. So it's like a integration. It's kind of a data integration where we have like fixed weights, which are mapping from the germline DNA to the tumor RNA. I'll have a slide that shows it more clearly. So that's what we wanted to do. But first, we were presented with these batch effects. So we started to look at the data, and this is work by Arjun Bhattacharya, Alina Hamilton, and then more recently, Achal Patel. And we started to look at the data, and we saw here are three densities for the three phases of the study. The densities describe, if you take the measurement of gene expression, so you have many samples and many genes. It's a matrix that's genes by samples. And you kind of center the data so that a gene has a mean of 0. So now you have a matrix where the rows have mean 0, and you look to see the median deviation per sample. So now we have a per sample measurement. Is it tend to be lower or higher than the mean? And what you can see is the raw data, the values for phase 1 and phase 2 were tended to be lower. Those samples had tended to have lower values in the matrix, in the raw data. And then the normalization that had already been applied when we started was called Insolver. It helped a little bit, but you can still see clearly there's a difference in distribution across the phases, just residual in the data. So that's not good. That will confound the study. There could be different, aspects of sampling. There were different aspects of sampling across the three phases. So now we have a mix of probably a technical distortion of the measurements, which is confounded with the phase, and we know there's also sampling changes across the phases. So this will be a problem if we just naively, go forward. Another way to see that is if we look at something we believe would be the most predominant biological signal in the expression of a tumor should be the ER status. So we can label the samples, whether the tumor is ER positive or ER negative. That's like the most, that should be PC1, explaining the difference in gene expression in a tumor, whether the tumor is ER positive or not. So here you can see PC1, I didn't label it. PC1 on the x-axis in the raw data is not the ER status, something else is, and PC2 is ER status. After normalization, it's rotated, but it's not there yet. So still something else is dominant rather than what we believe the largest biological variation should be ER status. And this kind of, you can't do this for every data set. Here we already have a prior belief that this factor should be PC1. And then finally, this is another measurement of this is called the silhouette width. And this is a per sample measurement of basically how close are the samples clustering by phase. So the silhouette width would be high when the samples are near other samples of that phase. And so you can see the raw data is the green violin plot, a lot of positive values, meaning that samples tend to be with their phase. And then the normalized data is the orange one. Still, a lot of those values are positive. The percent of positive values is still 46%. So most of the samples tend to be similar to their phase. So we haven't removed this phase effect of the data. So there's some indication that we can't just go ahead and use the data naively. Here's this sketch of what we want to do. So what we want to do is, instead of associating DNA, germline DNA or genotypes, this green box, we want to find associations between germline DNA and phenotypes, so survival outcomes in breast cancer. We want to understand the heritable aspect of why some women have, worse survival, other women have better survival. There's a multiple testing problem. Typically we don't have enough samples to do this kind of study. So we are underpowered to just look at every genotype. Not enough sample size to do this type of analysis. So instead, this is a data integration idea, which has been proposed in the past decade, which is to learn a fixed set of weights, which map from genotypes to gene expression. So we learn these weights, just penalized regression models, just because of the dimensionality. So we learn, how do the genotypes combine to explain gene expression? Because we think that gene expression is a good mediator for outcomes. So we just fix those weights and then build this thing in the middle, the predictive model of expression. That's just betas, essentially, mapping you from genotype to now a smaller dimensional space, which is gene expression. And then what we do is we impute the gene expression in the study that we didn't have it. So this is, on the right-hand side, this is maybe, 500 samples. On the left-hand side, this is going to be like 1 million samples. We didn't measure gene expression in 1 million samples, but we can use the betas to impute the gene expression. Now we're going from millions of genotypes down to thousands of genes. So big reduction in the number of features. And we believe that this is, these weights are informative. They have mechanistic explanation. It's, how does the SNPs change the level of the gene expression? Yeah.
So you're learning that relationship from a different database?
Two separate studies. So the study on the right is our study, CBCS, where we have gene expression for thousands of women, and on the left-hand side would be a large GWAS where we don't have gene expression.
But would it be necessarily a reasonable assumption to say the underlying distribution for that database would be similar to there would be any kind of bias?
Yeah. So the the regulation of gene expression probably is shared across people. And so, but that's an assumption. And that's an active area of research. It's like, is the gene regulation, are these predictive models portable? And actually, I'll come back to that. There's kind of a two-part answer to that. But we do end up building stratified models here. We stratify by self-reported race. And so that's one component. And I think there's a technical reason to do that. Yeah, I have a slide for that. So we did this. We did this this right-hand part. We built a predictive model for gene expression in tumors in CBCS in our study. And this is what if you plot on the x-axis the genotypes, so the SNPs in the genome from chromosome 1 to chromosome X. On the y-axis is the genes, location of the genes. Our prior belief is that there would be lots of things on the diagonal because genotypes tend to affect genes nearby. And I have a data point to show that. Is from a GTEx. It's a large project which looked at the relationship between genotype and gene in many tissues. And you can see the distance from most of the regulatory relationships where SNPs affect expression of genes. So the genotype, whether it's 0, 1, or 2, explains variance in the level of the gene expression across samples. They tend to be very close to the gene. So the distance is in millions of base pairs. So you can see most of these relationships are 500,000 base pairs, which is actually like the line in the plot of the whole genome. The whole genome is 3 billion. So the line is this very close relationship. But instead, we see all these gray pluses, which are relationships that are not nearby, but kind of across different chromosomes. Those could exist, but we didn't think that there should be so many. There were a lot of them. What we think instead is happening is this. We have different populations, and populations could be defined by genetic variance. Populations could vary across study phase. So recruitment could be slightly different across phases. The phases also have differences in technical variation because the machines changed, the technology improved over the many decades of the study, and that technical variation affects the measurements we make. So that induces this kind of false connection between genotypes and gene expression. So that's what we think is happening when we see a false association between genotype and gene expression, is that there's these confounding relationships. So I won't go into the method details, but we used an existing method for normalizing gene expression, which is called, this is kind of our workflow. How do we approach renormalizing this data? And we used a method called RUV. I guess it's in the next slide. Yeah, the green box. We used an existing method called RUV. RUV is remove unwanted variation. And this seemed to do pretty well for our purposes. It involves an iterative aspect where you have to choose the rank of the nuisance variation, like the dimensionality of the nuisance variation. So we kind of tune that based on our prior beliefs. Like, we believe that ER positive status should be the dominant variation in the data, things like that. And there's also another method here, which is if you have technical replicate samples across the phases, that's very useful. So if you're designing a study that's going to have genomic data, it's really helpful to have, here's like 3 phases of some data set. It's good to have technical replicates that are measured in each phase. Obviously, that's one of your best measurements for the phase effect.
Phase is the stage of the concept.
Oh, sorry. Oh, study phase. Sorry. Yeah, I said stage. Oops, I think I lost the Zoom again. I said stage is the stage of the tumor, phase is the study phase. Yeah, sorry. Okay. So again, I'm bypassing like what is RUV? It's somewhat based on factor analysis, but it can also involve these technical replicate samples. And we renormalized the data until we saw what we wanted to see, which was PC1 being ER status. We saw much nicer deviations, like these per sample deviations. We don't see shifts by phase anymore. And now the samples are not more likely to be with their own phase. So this is just like, you can do many of these plots, and we have a bunch of examples in our paper. There's another examples in this RUV paper, plots that you should use if you have multi-phase data sets, or even any data set that has like 100 samples, or any data set ever. Please look at the data, make these kind of systematic plots, box plots, PCA plots, because otherwise you're kind of just gonna get junk. And this is what we see after we removed it, after we use our normalization. So it appears that a lot of these associations were maybe spurious. So we have some, it's not to say that there should be no X's off the diagonal. Those relationships can exist, but we think that probably those were spurious. Okay. This is what we wanted to do, going back. We wanted to associate genotype with outcomes, survival outcomes. And there had been studies previously. So this is not breast cancer. This is a review paper about this method. So there had previously been GWAS for breast cancer outcomes that had not discovered any significant features or genotypes. And this is basically the idea of, instead of testing or trying to find an association with every genotype in the genome, reduce the information by these fixed weights. So we train these models for gene expression, and we reduce by combining SNPs with this just a linear model. So then on the right-hand side, you end up with fewer features to test, and they're at the gene unit. So we did this with tumor gene expression for 600 Black women and 571 white women. We broke apart the models by self-reported race. And I think the reason that we did that, I'll show you, but it's probably not because of the underlying biology, but it's probably driven by the differences in SNP allele frequency. So the differences in the allele frequencies are probably the reason why the models are best stratified. And we evaluate these predictive models with cross-validation, also permutation to try to assess the variance. We also have held out data in our sample, and we have data from other studies. So we kind of have three measures of predictive accuracy. There's cross-validation, held out data in our study, and external study. So this is the how well can we predict gene expression. So it's the R squared from genotype in the tumor. And there are some points in the top right, which say we can predict gene expression really well. There's one PSPHL. We can really well predict the expression in the tumor just from genotype. So if you think about that, we can predict the gene expression before the incidence of the tumor. So that's kind of an interesting concept. Like, if you just give me genotypes, I can impute something that doesn't even exist. What would the expression be in a tumor that doesn't exist? On the x-axis is the accuracy, this R squared in the white patients, and on the y-axis is the Black patients. And so we built these separate models and we looked to see, are the same genes predictable in these two populations? So, and this is in our study. So there's some shared, these purple, and you notice the to call something like we can predict it, this is a very low standard. 1% variance explained is these dotted lines. So that's a very low amount of prediction, right? 1% variance explained. But people have shown even at really low single-digit percent variance explained, you can still use that to gain power in these association studies. That's kind of surprising that like explaining 5% of a gene expression would help you somehow in association studies, but it is the case. This is in an external data set. So you can see many things disappear when we go to an external data set. And we don't really know why. The technology was totally different, could be one reason, but two things remain. PSPHL was not measured in that data set, but it would appear in the top right if it had been measured. And then this next plot is what if we try to explain variance using we crossover. So we train a model in the white patients and try to explain variance in the Black patients, and the models don't do that well. I think the reason is the minor allele frequencies are totally different. So something that has a high minor allele frequency in one population based on ancestry might explain a lot of variance, but if that allele isn't very present in the other population, it doesn't help you. So the models are based on both like the effect sizes, so the relationships of the SNPs to the expression, but they're also based on the allele frequencies, and those differ between populations by ancestry. And then the last thing I want to point out is this gene in the top right had actually been highlighted in breast cancer research as being very interesting because it is differentially expressed across if you compare self-reported race, it has a differential expression in the tumor. And so some studies had said, if you think about it, these women are having more aggressive tumors. Let's just look at the difference in gene expression by race. And so it pops up as a differentially expressed gene when you compare tumor gene expression from Black women and white women, but that doesn't mean has any predictive power for outcomes. And so what we see that it's very it's almost perfectly explainable by genotype, and the reason is that it's actually a pseudogene that probably doesn't do anything, and it has a genotype that explains its expression level very well. And those are it's basically just a marker of population, but it has nothing, just because we could show that if you stratify, it has no explanatory power between cases and controls, and it didn't help to explain outcomes at all. So this is as opposed to like just doing observational comparisons, working from germline can help to explain whether something is informative for survival or not. Let me see. I want to jump ahead because I'm coming up to the end, and I'm going to jump through the results we saw. There's, this work, this is called transcriptome-wide association study. So the idea of going from the genome to the transcriptome or the genes. This is like ongoing work, and the student is now postdoc Arjun Bhattacharya. He had done this analysis. He's now been working on going to higher level of resolution, looking at gene isoforms instead of just total gene expression. And very interesting work comparing haplotypes, comparing across chromosomes within admixed populations. So this ends up being probably, if you want to look at ancestry effects, comparing blocks of DNA in admixed populations is probably the best way to try to estimate if there is an ancestry effect or not. So there's a recent preprint that's really, I think, a promising approach to try to disentangle ancestry-based effects by looking within admixed individuals. So pieces of DNA so humans don't have, a single ancestry, but pieces of DNA have ancestry. Like pieces of DNA have passed through different histories through time. And so you can say, this is a piece of DNA that came through European ancestry. This is a piece of DNA from African ancestry, and you can make those comparisons within individuals. And that's probably the right way to do this. And then the last thing is a note that in general, we can't do this division of individuals into groups by self-reported race. There's clearly a continuum of ancestry in human populations. And so this is like methods describing better ways to do this inference on genotype for incidence and outcomes in disease, is to consider the continuum of ancestry. I want to share this concept of somatic mutations, but I won't get into our methods, but just kind of entice you into thinking about this as a possible type of data you might look at, and then I'll conclude. So now I'm focusing on a different subset of data in this study. We have risk factors. We have the mutations that occurred before the tumor, somatic mutations in the genome, and we also have mutations that occurred after the tumor. And with just those bits of data and some external data sets, we can figure out some key ideas about the etiology of the tumor. And so down here on the bottom, I have, there's a data set called COSMIC, which describes what types of mutations occur from what different sources. And so let me just explain that idea and then try to get you excited about somatic mutation data. So UV radiation induces mutations over time in the genome, in somatic cells. That's one type of exposure. This is on the right side, this is a mutation occurs in the tumor upstream of a gene. And this gene is an enzyme that is mutagenic. So you don't want this gene to be highly expressed because it starts inducing mutations in the genome. It's actually a self-defense mechanism, this gene here. So we have a mutation and that increases the expression of this gene. Now there's a lot more of this protein floating around, and that protein is creating a lot of mutations in the genome. So this is another type of mutation that we see. The first one is from exposure, and the right one is from after the incidence of the tumor. A mutation in the tumor is producing additional mutations, and that's tumors evolve to produce more mutations to have hypermutant status because they can evolve faster with lots of mutations are the foundation for natural selection, and tumors are trying to evade the immune system and things like that. So they want to have more mutations. And these mutations have particular patterns that they make. So UV radiation most commonly turns TTT into TAT. That's a very common mutation if you expose DNA to UV radiation. And so you can capture this in one of these diagrams below. So it's just a bar chart of all the 96 possible triplets of DNA with the middle piece of DNA changing from one letter to another. So that if you do the math, that ends up being 96, considering that we can't observe a T to a T. So there's only less than 16 transitions. That ends up being 96, and we can just see how often does UV radiation induce these different types, and that's kind of like that's called a signature. Like it's a pattern that is observed that points you towards the etiology of the mutation. This one on the right-hand side, for a particular enzyme that we see often mutated in cancer, is called ApoBec, and the mutations look like this. So it's just different types of patterns we see in the DNA when we look and we compare to exposures or mutated genes. So it's very interesting to discover these patterns. There's many techniques to kind of discover them de novo. So matrix factorization methods can look at a matrix and try to cluster the types, the patterns that you see. And then people want to associate those with exposures. So the ones that are not already, like UV radiation, I said, is already associated with an exposure. There are some of these that don't have an exposure. You see a pattern common across patients, you want to find an exposure. So you can estimate how much of a patient's mutations come from a particular pattern, and then you want to associate that with an exposure. So here, like this study wanted to associate it with race. So with women from the Nigerian study and women in the American study stratified by their self-reported race. So you see those bars, they're making comparisons across these amount of a signature 8. So how much of signature 8 was there? And the point we're making here is that those are actually estimated from the data, and then they forget the fact that it's estimated and just do a comparison. But it's estimated, and it's estimated often from low numbers of counts of mutations. So basically, the point of this last bit is that you should not ignore the fact that it's an estimated quantity, and we have instead a hierarchical model that we've been working on to try to make these comparisons. Okay, so I'll stop there just with the conclusion slide because I don't want to go too much. I want to like present my my recommendations for working with large data sets. So please look at the raw data if you work with these kinds of genomic measurements, or anytime ever. Look at data summaries across things like time that the sample was created, or phase, batches. Ask the collaborator, were the samples processed in batches, and can I have that variable when you send me the data, please? So this is really important, or else you're going to definitely run into mistakes. Those need to either be corrected for or like included in the analyses. Ask for positive and negative controls or pseudo controls. What do you expect to see? What should not show a difference? What should show a difference? And are you recovering what's known, or do you have to go back and reprocess the data because you're not seeing what you expect? If you don't know what those are, talk to the collaborator and figure them out. There should be something that they expect to see. And those, I think, are important for these large scale studies. Consider relationships of variables, confounders potentially interacting with exposures. And I just want to say, like, I showed a couple of studies and questions we've been trying to answer. There's huge demand for statisticians and people who can work with data. So it's a great time to be doing applied research. And I'll stop there. I want to acknowledge the participants in the study and the volunteers and the staff, the 2 labs that I've collaborated with, Troster and Hoadley Lab, and the students who led these works is Arjun Bhattacharya, Jiun Park, and the last paper I didn't get to talk about was by Sarah Reifeis. Thanks so much.
Thank you so much, Michael. It was a very interesting talk. I'll open this up for 2 questions right now. Yeah.
Thank you for the talk. So you discussed a little bit on how you address the batch effects associated with the cases. And then you talked a little bit on how to sort of disentangle ancestry effects from just comparison. So in your opinion, as of right now, what is maybe like the biggest challenge that is left? Sort of address some of them.
Yeah, well, there are cases where ancestry is totally entangled with the study, and it's very hard then to try to learn a model for example, like these predictive models for gene expression. It's very hard to kind of extract out the contribution of technical variation and then the kind of the aspect of the ancestry and the allele frequency. So right now, it seems like these kind of ancestry specific models are being built. Although I want to emphasize that there's this important work recently, the second one here, that it's probably not that the effect sizes are different, but it's because the allele frequencies are different. So if you were to put that allele in a different population, I think the effect would be the same. That's what actually shown on the right-hand side, the betas. The effect of the alleles are probably the same, but because the alleles have different frequency at different populations, that makes the models less portable. That's like a very promising topic and area for research if you're interested in genetics and genetic association.
Just to follow up, is it possible that from a beta model, you can split the allele frequencies from the together with the variance or the same direction? I mean, it's obviously going to be huge, but
Yeah. I guess there's another problem, and I didn't talk about this at all, but we don't measure all the alleles. We measure a subset, and the ones that we measure tag the causal betas, the causal allele. So that's a problem. So basically, like if we don't observe the tagging SNP, we have no hope to tag that to the causal SNP. That's the problem in these like polygenic scoring and also in these expression models being used for GWAS. Yeah.
So how connected is genetic ancestry to self-reported
In the CBCS, it was pretty much 1-to-1, but that's not the case in all studies. I think that has to do with the populations in North Carolina, that when we measured, we estimated percent African ancestry and compared that to the self-reported race identified from the clinic, and that was a 1-to-1 mapping. We could see there was a gradient of admixture, but that's just not the case in general. And this last paper, I think, talks a lot about that. So that the colors here are self-reported, and the axes on the x-axis is like by genetics, what's the distance between individuals in terms of genetic variance. And it's not a 1-to-1 mapping. There are people who identify where their genetic variance, say something. It's clearly a continuum.
So it's dangerous that you use like self-reporting
So the reason we did that is because we have these like very different allele frequencies.
Yeah.
So we build separate models because the models don't work across because of this issue of allele frequency differences. So if you want to build a good model, you have to use the alleles that are present in the population. And so it would have been identical to separate individuals by ancestry, by the allele frequencies. It would have given us the same separation in this study, but that's not generally the case.
So, why did you choose Nigeria?
Oh, and I should say that's a study that I didn't do, but I think this is from NCI.
Genetics variation in genetics is a lot more than the variation within, say, European genetics. So it's dangerous if you look at like one country, one tribe, and then map it over to the rest of the continent.
Absolutely. There's a Nigerian study. There's also a Ghana study. These are just scratching the surface, right? It's just one study. And so, yeah, this paper is, one study, but I would also encourage you to check out the Ghana study for breast cancer incidence and subtyping. And there should be more. We should be doing a lot more genetic studies in African populations. Yeah.
so if you are working with soup, then the confounding due to ancestry, is it as accurate as the germline mutation? Do I also need to look at like this confounding or just?
There's no confounding because we compare the somatic mutations to the germline. So we remove the germline variants. So we're just seeing the delta, and those don't reflect ancestry. These are new variants from the exposures.
Are there any other questions for Michael? Or if not, let's thank Michael again.